Welcome to Activity 5.3, How do nutrients move into a cell? In our lab, How Do You Eat a Cracker, we learned that a saltine cracker is a type of starch. And when a starch is mixed with your saliva, there's an enzyme called amylase that breaks starches down into glucose. This is a chemical reaction. What we are trying to understand is why this is a necessary step in the digestions of foods. Why do we have to chemically break down starches into glucose for your body to use? Why can't your body use starch molecules as they are? To understand this, we need to understand permeability. So go ahead and open your WordWall spreadsheet and type in these terms as we go through them. Both plant and animal cells have a cell membrane that separates the cell from the outside environment. A barrier could be characterized as permeable, meaning it has pores and openings that allow substances to freely pass through, or it could be impermeable, meaning it's a barrier that does not allow any substances to pass through. Both of these types of barriers would not be appropriate for a cell's survival. If a barrier on a cell or its membrane is permeable, then anything can get through, including bad things like bacteria and viruses. If a cell membrane were impermeable, bad things would stay out, but also the good things the cell needs to survive will not pass through, such as oxygen, water, and nutrients. What we need is for a cell to be what you call semi-permeable, that the cell membrane is something that allows some substances to pass through and some substances need to stay out. Cell membranes are semi-permeable. The method through which particles can pass into a cell is called diffusion, because during diffusion, molecules move from an area of high concentration to an area of low concentration. So if you have something like nutrients highly concentrated outside of a cell, they are naturally going to diffuse or spread out until they are evenly spaced and have even concentration with the inside of the cell and outside of the cell. So the purpose of this lab is we are going to test if starch and or glucose can move in and out of a cell. Therefore, we need to create a cell model. Our cell model has to have the same type of permeability as the cells in our body. Therefore, we need a material that is semi-permeable. Our cell model is going to be made from dialysis tubing. It seems like plastic, but it actually has microscopic holes just like a cell, and it is semi-permeable. To set up the lab, we wrote our period and our group number onto a cup with permanent marker, and then we filled it with water. Then we soaked the dialysis tubing and opened it up. We tied off one end and filled one half with glucose solution and the other half with a starch solution. And then we thoroughly rinsed it underwater to get rid of any remnants of starch or glucose on the outside of the tube. Then we put the model into a cup with water and we were waiting for 24 hours. After 24 hours, we are going to test the water in the cup to find out what substances were able to diffuse out of the dialysis tubing. Because it is semi-permeable and it's all concentrated in the dialysis tube, we would like to find out if starch and or glucose was able to diffuse out. So in your book on the lab, you are supposed to make a diagram. The diagram for day 1 is supposed to indicate how our model is set up. You need to draw particles for water, starch, and glucose. You would draw an empty dialysis tube and then draw in circles or particles to represent where is the starch and the glucose and the water after you set up your model initially. The next day, or after 24 hours, we need to test this water to find out if starch or glucose diffuse out. In order to test, we need something that's called an indicator. An indicator is a chemical that is used to detect the presence of another chemical. Therefore, we need an indicator for glucose and starch. Benedict's solution is something that can test for glucose. If glucose is present, it turns orange. If glucose is not present, it stays blue. For the test, we take a sample from the cup after 24 hours of water, and we put 5 to 7 drops of Benedict's solution into the test tube, and then we put it in a hot water bath. After some time, either the test tube is going to be blue, meaning there is no glucose, or it would be orange, which is a positive test for glucose. To test for starch, we need a substance called iodine. If the starch is present in the substance, it will turn black. If starch is not present, it would stay brown. The test is immediate. You just take a sample of water from the cup and add 2 to 3 drops of iodine into the cup's water. A negative test looks like this, where it's brown, meaning there is no starch. A positive test for starch looks like it's blackish purple. Students then submitted their findings in the shortened URL. The results were as follows. 93% of the students tested positive for glucose. Those who had a negative test might not have heated the solution long enough to see any change. And also, after 24 hours, we ended up with a negative result for starch. There was no starch present in the water. Students who got a positive might not have rinsed their cell model thoroughly, or it could have leaked overnight and they got a false positive. So you need to make a diagram on your back page that indicates the change after 24 hours. You need to draw in particles that indicate where is the water, where is the starch, and where is the glucose. Especially you need to think about where is the glucose relative to the inside and outside of the cell model. Is there still glucose in the cell model after 24 hours, or did all of it diffuse out? Then you have to answer the following question, where you have to explain what happened to the starch and glucose particles during the experiment and why this happened. Here are some things to help you out. All right, you need 2 word wall words. So here is a word bank for you. You need to use the words that we taught you in this video: semi-permeable, permeable, impermeable, or diffusion. You need to pick 2 of these words and use them properly in your explanation. In addition, for why it happened, you need to remember that starch is made up of glucose. So relatively, you need to think about what is a bigger molecule. Is starch a bigger molecule, or is glucose a bigger molecule? And why does the size of the particles determine what was able to go into the water after 24 hours?